Today the topic of our discussion is monoclonal antibody production by Hybridoma technology. At first we need to know what are monoclonal antibodies. So monoclonal antibodies, these are some lab-made antibodies which are specific for a certain type of antigen that are represented by the cancer cells. Now there are various types of monoclonal antibodies and each one of them is designed to attach to a single and specific type of antigen. We can say that monoclonal antibodies are involved in the targeted treatment because they only target a specific type of antigen. Here it is a very gross representation of a cancer cell which is having various types of antigen on its surface like the triangle, the circles, the square ones. And the monoclonal antibodies, they are specific for the circle ones in this case. So they will come and bind only to these specific circle antigens. For the production of monoclonal antibodies, we are using the Hybridoma technology. And to begin with this process, at first we need a mouse. We inject the mouse with some non-self antigen, and as a result of this infection, some B cells containing the specific antibodies are produced in the mouse. Now specifically these cells are produced in the spleen of the mouse, and we then isolate the B cells from the spleen. These B cells have the following characteristics. They are AGPRT positive, that means they produce the hypoxanthine guanine phosphoribosyl transferase enzyme. They are TK positive, that means they produce the thymidine kinase enzyme, but these cells are mortal in nature. That means these cells die after a few weeks. And as a result, we don't get a production of the monoclonal antibodies for a longer time. And for treatments, we actually prefer to have a alternative where we can get the benefits of the monoclonal antibody for a longer period of time. So here it is the problem with the B cells. They are mortal in nature. Now in Hybridoma technology, another cell is involved, that is the myeloma cells. These myeloma cells, they are AGPRT negative, TK negative. That means they don't produce these two enzymes. On the other hand, they are immortal in nature. So this is the characteristics we want in our B cells, so that there is unlimited production of the monoclonal antibody. In order to make the cell with the characteristics of being immortal, AGPRT positive and TK positive, we need to fuse the two cells so that we can get a single cell having characteristics of both the cells. Now the fusion of the cell of these two cells is performed in the presence of the polyethylene glycol. Now this is the agent which fuses the plasma membrane of two adjacent cells and make it a single cell containing multiple nuclei. Now the fusion products, there are various possibilities of a fusion product, okay. So it may occur that the two B cell has fused together, two myeloma cells may have fused together. Hybridoma is the one that we need, the fusion that we actually aiming for. Or it may occur that the myeloma cells or the B cells, they may not have fused. So you can see there are 5 types of possibilities that are possible upon fusion of the cells. And from these possibilities, we only need the hybridoma cells. These hybridoma cells, as I mentioned, have the characteristics like they are AGPRT positive, TK positive, which are attained from the B cells, and they are immortal in nature, and this characteristics is attained from the myeloma cells. The advantage of the hybridoma cells is that there is unlimited production of monoclonal antibodies. Now, in order to separate out the hybridoma cells from the mixture of the cells, we use the HAT media. Now, HAT media stands for hypoxanthine aminopterin thymidine media. Now, before understanding the procedure of the selection, we need to clear our concept about de novo and salvage pathway. Before a cell divides into two daughter cells, the parental cell undergoes replication of its DNA. And during the replication, we know one strand is parental and another new strand is formed in complementary to that parental strand. The production of the new strand requires the synthesis of nucleotides, and synthesis of nucleotides are possible by two pathways. One is de novo pathway, another one is salvage pathway. De novo pathway is the most preferred path for any cell, and in case the de novo pathway is not working, the next preferred pathway is salvage pathway. Now in the hat media, we know we have aminopterin in it. Now aminopterin is known to interrupt the de novo pathway, and as a result the nucleotide synthesis is interrupted. But as I mentioned that in case the de novo pathway is not working, the cell will use the salvage pathway. Now for using the salvage pathway, the cell needs to have HGPRT and TK enzymes in it so that they can utilize the hypoxanthine and the thymidine that are present in the hat media, and they can perform the nucleotide synthesis. So here we have the hat media containing the various types of cells, the various possibilities that we earlier discussed, the 5 types of cells. And here the B cells, both fused and non-fused, they won't be able to survive in this hat media because they are mortal in nature. Although they have the HGPRT and TK enzymes in them and they can synthesize their nucleotide by the salvage pathway, but still they can't survive in this media because they are mortal in nature. Next we have the myeloma cells. Now both fused and unfused, both type of myeloma cells, they are immortal in nature, we know that, but they are HGPRT and TK negative. That means they don't have these enzymes in them, and we know that already the de novo pathway is interrupted because of the aminopterin that is present in the hat media. So the next preferred path was salvage pathway, and these cells don't have the HGPRT and the TK enzymes in them, so they can't perform the salvage pathway either. So as a result, they will die. Now only the hybridoma cells, they will survive in the hat media because they have the HGPRT and TK enzymes in them, so they can continue their nucleotide synthesis by the salvage pathway, and also they are immortal in nature. So as a result, only these cells will survive. The next step after selection of the hybridoma cells is that determination of their specificity and bulk production. To determine the specificity of the hybridoma cells that producing the antibodies, we perform the sandwich ELISA. And from the sandwich ELISA, we get a chromogenic response which helps to identify that how much specific and potential our antibody producing colonies are, and we can validate them and characterize them, and we can isolate those specific potential colonies in a separate chamber, and we can scale it up in larger flasks, and then we can use it for targeted treatments. So this is all about the monoclonal antibodies. Thank you for watching and subscribe for more updates.